Okay, we're going to get started. Good morning, good afternoon, good evening, depending on where you're joining us from. On behalf of the Protein Society, welcome to today's webinar entitled Structural Biology of Trip Channels at Different Temperatures. I'm Heather Pinkett. I'm president of the Protein Society, and we're thrilled to have you join us today in what promises to be an exciting and insightful webinar. Before we begin, I'd just like to take a few minutes to present some updates. So first and foremost, let me do slide change. Awesome. We're excited to announce our next annual symposium that's going to take place on July 19th to the 22nd, 2026 at Westin Boston Seaport. This is a great opportunity for you to catch up on the latest cutting edge science in the area of protein science. We have a series of wonderful talks and interactive poster sessions, and it's a great opportunity for you to catch up with the scientific community. It is the highlight of our scientific calendar. We hope to see many of you there. We also encourage you to engage in our journal, Protein Science, which serves as our official publication of the Protein Society. It's a platform for rigorous, high-impact research in all areas of protein science, and it includes our tool issue that's coming out in January. So I hope you're able to catch that. If you're considering submitting an article, now's a really great time to explore what the journal has to offer. And so, as always, none of this could be possible without the dedication and leadership of the Protein Society. And so a heartfelt thank you to our past president, Liz Meering, who led the charge on webinars this year, and Shannon and Raluca for their tireless work behind the scenes on the webinar and supporting our society year-round. Now for some logistics. So today's webinar, each speaker is going to present for 25 minutes, and there'll be 2 to 3 minutes for questions after. And at the end of the webinar, when all of the talks are done, we're going to then have a discussion. So that's another opportunity to introduce questions. So please place any questions you have in the Q&A tab, and the organizers will read them out loud. Now I'm delighted to turn it over to today's organizer and moderator, Dr. Yifan Cheng from UCSF. Dr. Cheng is a leader in the field of membrane proteins and structural biology, and we're fortunate to have him guide us today. Yifan, I'm turning it over to you.
Thank you. Can I share the screen now? Yeah. Can you see my screen? Can you see my screen? I can't hear.
Not yet.
Now?
Not yet.
Yes, can you see my screen? Or
No.
I'm trying to share screen. Oh, okay, share.
Not yet. Go
Now
ahead.
it's it. All right.
Now we see it.
Thank you, everybody. I assume you can see my screen now. And my name is Yifan Chen. I'm from UCSF. I'm the host of moderator of today's webinar. And then, so I just want to take a few minutes to have a brief introduction of today's theme. The title of today's webinar is Structural Biology of Trip Channel at different temperatures. Here, there are two keywords in this on the theme. One is the trip channel, and one is the temperature. And the trip channel is a large, diverse, like a superfamily, and it stands for transient receptor potential ion channel. And it's in normal, it has 6 subfamilies. And then all these channels perform very different cellular functions. And they all share some common feature in the transmembrane domain, but then their function are very different. And then some of them, such as in the TRP-V family and the TRP-V1 and 2 or 3 and this one, and also some in the TRP-M family like TRP-M8, They are in the sensory neuron and then sensing the environment temperature and the response to those temperature. And some others that work in the other part of the body, and then they maintain functionally critical in a certain specific physiological temperature. In terms of the temperature, that's the one thing that for some of TRP channel, they respond for the temperature. And such as in the TRP-V family, there are some channels that respond to the temperature and then generating the pain sensation by the heat, some other by the cold, generating the pain sensation. So you can see from this trace that this is an early paper, shows that the channel, how the response to temperature. So one of the theme for this webinar is to look into structural biology, how does this channel is being regulated by the function. The other part of the question about the temperature is that we all there are more and more data supporting the idea that the channel function and then more specific at a certain specific temperature. This is one example, not in today's covering this ion channel, and I think this channel also Jen's lab working on. And then this channel, you can see that it's only got a current at a 37 degrees C, but not in the room temperature. Suggests that a study channel at a specific physiological temperature is an important point. So that's why today's webinar focus on the TRP channel structure and also related to the temperature. And then this is also possible nowadays with structural biology in the era of single body cryo-EM. It is much more feasible. It's not say easier, but more feasible to capture structures at a different temperature. For example, you can maintain the temperature when the sample stay on the cryo-EM agrees before you plunge freezing. And then the cooling rate of plunge freezing is about like 100,000 degrees C per second. Such a high cooling rate will probably allow one to capture the structure at a specific temperature that was before the plunge freezing. And then so today's we have a lineup for 3 speakers. And our first speaker is Sasha from Columbia University. She will talk about TRPV3, which is a heat-activated channel, and then followed by Jian from Northwestern University. And I believe she will be talking about TRPM4, then followed by David Julius from UCSF, and he will talk about the TRPM8 channel. Now I'll turn into our first speaker, Sasha. Please go ahead, Sasha.
Okay. Okay, can you hear me?
Yes, we hear you and see the slides. Go ahead.
Okay, well, first of all, thank you, Yifan, for a nice introduction. And thank you, organizers, for inviting me to present data from my lab here. Well, when we look in a window, right, we all know when it's cold or hot outside. But not only we see and know about that, we actually feel it, right? And one common way, like one usual way to actually talk about that is, common example, if we touch something cold or hot, we feel it, right? Because our sensory neuron brings information to the spinal cord, then synapse on the motor neuron, which brings information back to the muscles and generates a reflex reaction. So what is actually happening here at the place where we touch caught a hot object? In the skin, we have a number of ion channels which are sensitive to changes in temperature. They open their pore and conduct current in response to changing temperature, and that generates action potential, which travels to the spinal column and synapse to either motor neuron to generate reflex reaction or brings information to the brain so that we actually know that we touching hot or cold object. And today, as Yifan already told you, we are gonna discuss the primary perceptors of hot and cold sensation, the ion channels. And many of them belong to this family of TRP channels, which cover a broad range of temperatures from noxious cold to noxious hot. And they're also interesting because they're regulated by small molecules that in humans are associated with senses of taste and smell. And of course, a few years ago, these 2 gentlemen were awarded a Nobel Prize for discovering these channels, including David, who is gonna speak later today. So, of these 7 subfamilies of TRP channels, as Yifan already said, we're gonna be talking about different representatives, and I'm gonna focus on TRPV3. TRPV3 belongs to vanilloid subfamily TRP channels, mostly expressed in skin and few more organs, but physiological function, as you can see, mostly related to skin physiology. Accordingly, a lot of diseases related to TRPV3 are skin diseases like psoriasis, rosacea, acne, but also this genodermatosis called Olmsted syndrome, where a single mutation in a TRPV3 gene can cause very severe consequences for human skin. This channel also related to several human cancers and represents important physiological and pathophysiological target. One important and interesting, exciting property of this ion channel called sensitization. So in function, we can see it in this example where whole cell recordings are made from HEK cell expressing TRPV3 channels, recording made by the lab of Vicky Vlacheva. You can appreciate repetitive stimulation of this heat first induces small current through TRPV3, which gradually increases in amplitude. So this is exactly opposite to majority of ion channels which desensitize or inactivate in response to repetitive stimulus with a decrease in current response. Sensitization of TRPV3 doesn't have to be monitored by in response to repetitive stimulus. It can be seen in response to continuous increase in temperature as an experiment shown here. Here is a completely different setting. The TRPV3 protein was first purified and then reconstituted into lipid bilayer, work done in the lab of Ella Zacharian. And you can appreciate as the temperature goes up, first all channels are silent and then more and more channels start to fire. This experiment can be repeated if instead of many channels, only one present in lipid bilayer, then we obviously have single channel recordings, example of which is shown here. At 25 degrees, we have no activity of TRPV3, then it's increasing at 36, it appears at 36 and getting more at 42 degrees. So the current voltage relationship is close to linear, while the temperature dependence is very non-linear, and you can appreciate the open probability of the channel remains close to zero till temperature reaches about 38 degrees, and then the further rise of temperature causing sharp increase in open probability, causing this higher temperature coefficient, the signature of temperature sensitive ion channels. And my lab is structural lab, so we wanted to know how protein responds to the temperature. And these two gentlemen, Kirill and Arthur, they purified some of these, they expressed and purified TRPV3 and then reconstituted this channel into so-called lipid nanodisk using nanodisk protein and lipids. And then they applied the purified sample onto cryo-EM grids and inserted it into cryo-electron microscope. There, electrons pointed at the sample create a picture on the detector called electron micrograph, which is example shown here with TRPV3 particles circled in red. And if we average bunch of them, we would get these so-called 2D class averages where you can already better see the shape of the molecule and start seeing some secondary structure features. And of course, this 2D information can be converted into three-dimensional maps, which is shown here. And we've been lucky enough because this reconstruction in MSP2 and 2 nanodisk gave us pretty high resolution reconstruction. You can appreciate holes in aromatics, water. The quality of the map where it's pretty easy to build a molecular model, which is shown here. And those of you who've been staring at TRP channel structures know that this is typical vanilloid subfamily TRP channel, which has this characteristic intracellular skirt, mostly built of ankyrin repeat domains, connected between themselves with these three-stranded beta sheets. In the TM, we have architecture of the ion channel typical for tetrameric ion channels, with the core of the channel built of two transmembrane helices and reentrancy p-loop. And on periphery, we have these four tetrahelical bundles called S1, S4 domains, which in voltage-gated channels play a role of voltage sensors. And the helix, which runs nearly parallel to membrane, called TRP helix, is a signature of the family. Okay, so we got this channel. Obviously, we wanted to know temperature dependence of this channel. And we subjected it to control experiment at 4 degrees. And we processed data and found only one population of particles. We call it closed. Why? You can see when we look at the pore, it has two constrictions. And the lower one is formed by this isoleucine 674 hydrophobic residues, most likely creating hydrophobic seal. To make sure, we collaborated with a group of molecular dynamic simulations done by Roman Efremov's group. And you can appreciate confirming our guess. Neither water, which is blue mesh here, nor ions, which is red mesh here, conduct through this ion channel. So it's truly closed. Okay, now we subjected our channel to a train of heat stimuli. And when we processed data, we found two populations of particles. We call them closed and sensitized. And as you can guess, the pore is getting dilated here compared to the closed state. And we almost been very happy and thought that's it. We got the open state, but our collaborators disappointed us by saying, no, water and ions still don't go through the entire pore because there is a hydrophobic seal impermeable to them at the isoleucine 674. Okay, then we've been looking for tricks how to make it work, and we found the trick was finding alternative nanodisk protein. This is CNW11 circular nanodisk. Again, at 4 degrees, control experiment gave us what we call closed state structure with no permeation of ions of water. And then when we subjected this protein to heat stimuli, again, we got two conformations, two populations of particles, which resulted in closed, and now we call it open. And well, you can guess why, because the pore here is much wider than in closed state. And of course, when we run molecular dynamic simulations, they showed that both water and ions freely go through this open channel. So we finally got the opening of the ion channel in response to heat. So I showed you four different conformations, what I called closed at two different nanodisks and two different temperatures. And this is superposition of individual subunits. You can see how similar they are, as well as their pore profile. So at the end of the day, our structural experiments with mouse TRPV3 resulted in three different conformations, which we called closed, sensitized, and open. With increasing size of the pore, and two of them non-conducting and one of them conducting ions and water. So what are structural changes are associated with heat? They already kind of illustrated here. So the N and C termini are highlighted. In a cold state, C terminus coming out of the last beta strand of the beta sheet wraps around this beta sheet, creating a glue between two neighboring subunits. So this cleft is filled up with the C terminus. What happens upon heating, this C terminus unwraps and forms a short alpha helix with completely different types of contacts to the neighboring subunit. And that opens up the cleft between two neighboring subunits, which suddenly and amazingly filled up with another polypeptide. And thanks to high resolution structure we have, we can trace it, we can build every residue in this polypeptide and determine this as an N-terminus of the neighboring subunit. So this interesting dance of termini upon heating of TRPV3. These are not only changes. If you look at the lipids, and focus on lipids which belong to one subunit, the upper leaflet looks pretty much similar, but obvious changes are happening in the lower leaflet, especially the one that there is a lipid which we labeled with V. There is a nice cleft formed in a protein which is fitted by this lipid in a cold state, and then this cleft completely disappears upon heating. And V stands for vanilloid. So vanilloid site was first structure discovered by Yifan and David Julius in 2013 in a structural work on TRP-V1. And in the apostate, this site is occupied by phosphoinositol lipid. And this is the site where agonists like RTX or capsaicin bind, or in fact, competitive antagonists like capsaicin. And so when ligand bind, you can appreciate the phosphoinositol lipid is actually getting kicked out of this site. Amazingly, the same kind of thing happened in TRPV3. So I will illustrate this by two different ligands. So these are different agonists of TRPV3. So if you look at this natural agonist from cannabis, right, this is actually non-psychoactive compound and has very useful properties like neuroprotective activity or appetite suppression and anti-cancer properties. So this one acts as an agonist at the TRPV3 and allows calcium through this channel enter the cell. So we solved the structure of TRPV3 in presence of THCV and found 4 sites, 1 per subunit. And it's amazingly, it binds to the same vanilloid site and, of course, competes with the lipid. The second example, which I show you, this is synthetic agonist 2-APB, the molecule which regulates different ion channels being antagonist or agonist. In this case, it activates TRPV3 and increasing concentration of 2-APB gets more and more calcium inside the cell. So we solved the structure. So we labeled, if we label vanilloid site number 1, 2-APB actually binds to completely different sites. And one site, we call site 2, it's at the base of the S1, S4 bundle. And site 3, it's below the level of lower membrane leaflet, below the TRP, the border of TRP helix and the so-called linker region between ankyrin repeat domains and the TM. And if you look at the vanilloid lipid sitting in the apostate in its pocket, right? Upon binding of THCV to the same site, obviously the lipid is getting replaced by THCV. But what was amazing, actually, the 2-APB binding to completely different site does the same. So you can see that lipid is gone from its site. So about the nature of the lipid, despite the pretty high quality of the surrounding protein, the lipid density, which is a black mesh here in our structure, doesn't look very definitive to say what exactly lipid is that. And so what we've done, our collaborators run MD simulations by introducing different types of lipids into this pocket and essentially seeing whether the lipid stays there and whether it mimics the experimental density. And of course, cholesterol looks very different in a predictive density, but the majority of other lipids stay in the site just fine, and many of them show shapes which are quite similar, like phosphatidylcholine or phosphatidylserine can easily be sitting in this pocket. So essentially, no definitive answer about specificity we got from these experiments, and this is something to figure out in the future experimentally. So apart from 2 dramatic changes, I showed you with termini and the lipid occupancy, overall, the channel during temperature transition is undergoing quite substantial conformational changes, which are illustrated by this movement of this curve towards the membrane by about 5 angstrom and rotation by about 8 degrees. And so pretty large portion of the channel is involved in these conformational changes, which we really call conformational wave because it involves so many regions. And we can trace this conformational wave, for example, starting with the vanilloid site where the lipid dissociates, and when it does, it changes conformation of the neighboring S2, S3 linker, which actually changing its helicity from N to C terminal part. And also dissociation of lipids moves the S4, S5 linker towards S1, S4 bundle. That in turn moves the entire S5 helix, and then the neighboring S6, which is closely related to S5. S6 in turn has gating residues, which allow to open the channel for conductance of ions and water. And S6 movement of S6 also translated to movement of neighboring TRP helix, which in turn moves the C-terminal hook adjacent to the three-stranded beta sheet, and there the whole dance of N and C termini happens, which I described you before. So this interesting experiment, so in a large part of the channel is involved in conformational wave, and that explains to some extent the previous mutagenesis experiments where people introduced mutations in different parts of the channel, and those mutations affected temperature sensation. Essentially, if you introduce obstacles at different locations of the wave, that obstacle can stop the wave independent of where exactly the obstacle is. But that also illustrates that, and that doesn't mean that that's the place where the wave is generated. Therefore, mutagenesis experiments seem to be deficient on the purpose of identifying the temperature sensor in these ion channels. Now, to describe kind of these changes in more detail, we actually worked with this. So I showed you actually 3 different states, right? Closed, sensitized, and open. In essence, we can break the temperature dependent structural changes in 2 steps, right? Closed to sensitized and sensitized to open, because sensitized state is somewhat intermediate. And one thing we've done, we measured RMSD with a short 10 residue window sliding through the structure, and this very much highlights local secondary structure changes in the protein. And you can appreciate S2, S3 linker is getting highlighted, and in C termini getting highlighted for the closed to sensitized transition. Interestingly enough, for the second transition, so nothing like this happening. And given that close to sensitized and close to open transitions are highly temperature sensitive, we actually think that close to sensitized transition is highly temperature sensitive, while sensitized to open is weakly temperature sensitive. Then, as a measure of structural movement associated with change in TRPV3 conformational state, we also calculated C-alpha atom translations between the corresponding structures which were superposed based on S1, S4 bundles. And these measurement of translation highlighted, of course, the regions where we observe secondary structure changes, S2, S3 linker and NNC termini, but also regions adjacent and which actually actively move as rigid bodies during these transitions. And this includes the entire ankyrin repeat domains part of the intracellular skirt. And so this is the pattern observed in a close to sensitized transition. And interestingly, for sensitized to open transition, we only have changes concentrated essentially to the gate adjacent area, which includes the C-terminal part of S6, N-terminal part of TRP helix, S4, S5 linker, and a little bit of the 4 helix. So these changes are illustrated in this movie. You can appreciate during the first step, we have the skirt going up, forming the C-terminal helices. During the second weakly temperature-dependent step, we have action concentrated to the pore gate, which opens and closes up for conductance, and then returning back to the closed state, unwrapping of C-terminal helices. Now we're going to look along the axis of four-fold rotational asymmetry. During the first temperature sensitive step, we have this rotation of the skirt, formation of C-terminal helices. Now, in the center, very important functional step, opening and closing of the ion channel, iris-like movement. And then returning back, again, rotation back of the skirt and dance of N and C termini I told you about before. All right, so at the end of talk, so what I told you about today, we succeeded to take a TRPV channel, TRPV3, and use exclusively temperature stimulus to go from closed to open state. We break it into two steps. First is close to sensitized transition, where we have dissociation of vanilloid side lipid and conformational changes in S2S3 linker and in C termini and movement of the entire skirt. The second step is weakly temperature sensitive and involves mostly movement around the gate region, and that opens the channel to conductance of water and ions. And of course, a lot of things not understood, and a lot of physical principles underlying these conformational transitions remain to be discovered. And some of recent work you will hear in the two following talks. But at the end, I would like to thank people who actually have done work. Most of what I showed you today was done by Kirill and Arthur. Apu and Luke started work with TRPV3 in my lab. Other members of my lab, our collaborators, labs of Vicky, Ella, and Roman, different cryo facilities who helped us to collect data, funding sources, and I would like to thank you all and happy to answer your questions.
Wonderful. Thanks, Sasha. We have a few questions here, and just a read out. And one of the question, the first question is why the TRPV3 conformation is sensitive to nanodisk formation, which is also related to my question about the sensitized states. If I understand it correctly, that when you using a bigger nanodisk, then you're able to capture the open structure. Well, I wonder whether that the sensitized state is actually really constrained by the size of the nanodisk. That's also related to the question asked by an audience with a initial AV.
Yes, thank you for the question. It's excellent question, and yes, like what we do in structural biology, right? It's often depends on the conditions we choose, and very often that leads to either breakage of a protein or fixing it in a specific conformation. That's why it took some time to actually vary conditions and see where the channel actually can behave and move close to the physiological condition. And I think, Yifan, you're right that the MSP2 and 2 nanodiscs, they create more tighter environment, more tighter nanodisk, and that doesn't really allow the channel to fully explore the conformational ensemble possible, right? While CNW11 nanodisk are more roomy and allow the channel to really experience the full transitions to the open state. And in that sense, sensitized state, what we call sensitized state, might be somewhat artificial, but it's still intermediate between the closed and open.
That's great. And then there is another question from the audience, and also related to one of my own questions, thought about it, is that what's the kind of your thoughts on how does temperature actually drives the this transition, conformation transition? It's a really challenging question because for me, I also remember that you in one of the slides you showed that you measured like I think the heat capacity change, right? Delta H, delta S, and then that may give you some thermodynamic readout. And then the question is really related to how does this thermodynamic sort of property drive the conformation change? Thanks.
Well, that's another excellent question, of course, and there are a number of hypotheses about how that happens, right? That's why at the end of my talk, I actually said that the physics of these processes are not quite clear, right? And one of the hypotheses was this heat capacity theory, right? Mentioned by Yifan. And by calculation, really, like if we calculate the number of residues which change their environment from water exposed to membrane exposed, right? Just by counting, it's sort of approximately right. So that theory could potentially work. But also the heat sensor theory is also cannot be rejected either. So it's something which we still, I think, have to work on and really figure it out at the end of the day, right? My favorite hypothesis is that the annular lipids could be working as sensors, right? Because we know that lipid membrane undergoes phase transitions, right? So this is abrupt change in physical properties in response to temperature. So those characteristics could dramatically change as you approach the channel closer and closer, right? So the lipid there can behave differently, but also react to as a phase transition in response to temperature. And that what would explain different behavior of different representatives of TRP channel family. And that also, of course, is in consensus with observations made by your groups and ours and others that lipids are actually contributing actively to the activation process of these ion channels.
Wonderful. I will ask one more question before we turn over to the next speaker. And this again is also an interesting question related to what also my own question is that the question is about like the flexibility and then the thermodynamic thermal sensitivity is the chart because the structure looks very well defined as I showed in the beginning, and then the high resolution, but that's also indicating the structure is very stable. And how does this stability of the channel that really relate to the thermal sensitivity of kind of your source? I think that's also related to my own question. And then if you cannot really we address this one fully, then towards the end we can also go back to this question to discuss of those three speakers. Thanks.
Sure. Yeah. Well, we using the structural method, right, of cryo-EM, and essentially, of course, nowadays we have some developments which allow to look at the dynamics of this protein, right, and that's something which we should be looking more and more. But the classical single particle cryo-EM, right, basically describes the stationary conformations, right? We select pools of particles out the all possible structural ensemble, and that's what we present as structures. But actually, if you look at the pool of particles, like at high temperature, for example, you have many particles which are not contributing to reconstructions, and those are reflections on the flexibility of the protein, which I think Yifan is talking about. So yeah, so this is one of, I think, of future directions is actually pay more attention to intermediates, to transient conformations, and try to characterize them. And those in fully kind of like as full as possible to describe the dynamics of the protein. Besides, that's not only that, even in stationary conformations which we reveal, big chunks of the protein are actually non-visible, right? So N and C termini, some of the loops. So yes, the dynamics is always there, and we have to kind of, while we're presenting this beautiful stationary structure, we always have to think about it. Thank you for the question.
Wonderful. And let's thank Sasha. And now we turn into our second speaker, Juan. So Juan, now it's your turn. Please go ahead, share your screen. Yes, everything's in order. Please go ahead.
Great. I assume you can hear me well. Beautiful talk, Sasha, and also thanks, Yifan, for organizing this wonderful event. And thanks also for all the audience for attending and listening. You are listening, right? Because it's a webinar, it's always hard to tell if you are sleeping or listening. I just assume you are listening. And this webinar is free. But if you know me, I never give a talk completely for free. And because this is a perfect audience, you have to let me do a commercial before my talk. I would love to advertise our upcoming channel receptor and transporter symposium at the 70th Biophysics Society annual meeting next February in San Francisco. As you have seen that we have put together a fantastic lineup of speakers, and I also encourage you to nominate a co-awardee. And the due is October 25th. You can see all the details on the BPS website. And I designed the t-shirt for our symposium with a Northwestern purple. So now is a perfect time to renew your BPS and CRT membership. Buy a co-award dinner ticket. It's going to happen in a beautiful Peruvian restaurant with almost unlimited wine and seafood. And also good time to grab a CRT t-shirt to show off. Okay, thank you for allowing me to do this. Of course, you can't stop me. Now I will go back to my talk. So here I show you a flow chart with the steps involved in determining the structure of a protein purified from recombinant expression system using single particle cryo-EM. And the process starts with protein expression and purification, and followed by cryo-EM sample preparation and imaging. And so you will generate the 2Ds, and the particle will be aligned and averaged, and then back projected to a 3D map. You can see the principle is really simple. Here I'm showing you a freezing protein in blue because historically the isolated proteins for cryo-EM studies or many biomedical and biophysical studies have been kept at a low temperature or room temperature to, as you all know, to maintain the protein integrity. We want to keep the protein happy. But we all know temperature is very important for protein function. That's why we are here now. So you can imagine in human body at body temperature, proteins are like people sitting comfortably in their home, nice and warm. And when they receive the red signals, agonists, antagonists, modulators, is like getting their version of trees. They will eagerly spring into action and being functional. And think about this when we dispatch scientists extract those proteins out of their comfortable zone and put them into coldness. Do you think they are still happy? So some proteins are really tough. They can handle whatever changes, but some proteins, they are sensitive. So if it were not their physiological temperature, they may slow down or even stop working. Think about yourself in December in Chicago, someone handed you ice cream if you want to take it. It's like, so now regardless how delicious those treats are, they may just not interested in taking them. And so today, I will talk about our work on trypan-4 as a notable example. And I'm not going to talk about temperature sensing, but I will show you why temperature may matter and how it potentially leads to interesting discoveries. And this work highlights the importance of physiological temperature for biomedical and biophysical studies and drug discovery. A brief introduction about TRPM4. TRPM4 belongs to TRP superfamily, melastatin subfamily, and is broadly expressed throughout the body. So it contributes to key physiological processes, including cellular depolarization, cardiac function, and immune responses. So these insights has been made possible by a lot of foundational and pioneering work of many labs. I just list some of them, and not all. It's too long to list everybody. And it's a calcium-activated non-selective cation channel and mediates cell membrane depolarization. And its activity can be modulated by small molecule compounds. For example, the voltage dependence of TRPM4 can be modulated by decavanidate or a short name DVT. You will hear this word throughout my talk. It's a metal cluster that carries 6 negative charges. And here in the E-phase recording, you can see a significant increase in currents at negative potential when DVT is present. And this results in a linear IV curve in comparison to the current without DVT. And another example is cytosolic ATP, which inhibits TRPM4 with IC50 in the micromolar range. The black trace here is current induced by calcium, and the orange trace is inhibition by ATP. So in 2017, we and other groups determined the first TRPM4 structures using sample prepared from 4 to 18 Celsius. And for your information, this temperature range is very typical for cryo-EM sample preparation. And several ligand binding sites were identified by different groups. In the transmembrane domain, Yifan, Cheng, and David Julius labs reported the agonist calcium site that is essential for channel activation. And you can see the residues marked in red containing the calcium TMD are absolutely conserved across calcium-sensitive TRPM family members, not just in TRPM4, but also in TRPM2 and 5 and 8. So if any of these residues is mutated to alanine, you can see this red trace is a wild type, and all the other colors, they are the mutants. So the channel no longer responds to, the mutants no longer responds to calcium. So binding of calcium to this site is indispensable for TRPM4 activation. And in the intracellular domain, our group identified 2 binding sites for DVT, if you remember, the positive voltage modulator for TRPM4. And also in the intracellular domain, Yuxin Zhang's lab at UT Southwestern identified a binding site for the inhibitor ATP. And altogether, these structures provided massive information about the overall assembly of the TRPM family. For your information, TRPM4 was the first structure determined in the entire family, and also ligand binding sites in TRPM4 at low temperatures. But very disappointingly, none of the TRPM4 structure was captured in an active open state. I think this is presumably because TRPM4 is voltage dependent and has high open probability only at positive membrane potential, which we don't have in cryo-EM experiments. However, it's still really puzzling that even we added a DVT, if you remember, the positive modulator that can render TRPM4 voltage independent, the pore remained closed. So we have struggled for a couple of years by trying different strategies to get an open state. I know some people here study structure function of ion channels, you know how painful it is if you cannot capture active open state of ion channel. We tried different detergents, tried nanodisk, tried different positive modulators, but they all failed. And eventually, we remembered an important feature of trypan-4, which is temperature dependence. And here we all know that sensation of temperature in humans and as a vertebrates is primarily mediated by members in the TRP superfamily. And in the TRPM family, you can see more than half of the family members can be activated by or directly activated or modulated by temperature from cool temperature to hot temperature. And here you can see the temperature sensing, their temperature sensing has been reported to be involved in different physiological functions, such as limits fever, enhanced taste sensation, and pain sensation. Here I just list a couple representative publications. So you can see among them, of course, TRPM8 is probably the most famous member is activated by cool temperature and also menthol. And although TRPM4 is not a famous temperature sensor like its sibling TRPM8, that guy got Nobel Prize. Oh, sorry, David, I forgot you are here. Pretend you are not seeing this and don't tell Arden. So TRPM4 has never been treated as a famous temperature sensor, but I will show you it still has pronounced temperature sensitivity. As you can see, it shows higher activity at physiological temperature here. On the left is the E-phase carried out at room temperature, and on the right is 37 Celsius. So I use a snowflake to indicate room temperature or lower, and sunshine indicate 37 Celsius. You will see those symbols throughout my talk. And so this reminded us why don't we study its structure at a physiological temperature using cryo-EM, where TRPM4 shows the highest activity. This may sound strange, but it's feasible, as Yifan mentioned, and as Sasha also done, because freezing process of cryo-EM sample is very rapid. So it's possible to preserve, at least partially, temperature-dependent protein dynamics on the grids. And so this work was mainly done by talented postdoc Jinghong Hu and also a senior research scientist in our lab, Dr. Songjin Park. And here I show you the video for the whole sample preparation process. Jinghong still purified the protein at a low temperature. Then he preheated the sample to 37 Celsius. And this is a virtual bot to prepare cryo-EM sample. This is great. And Jinghong kept the virtual bot chamber to 37 Celsius and this liquid ethane to freeze grace. And Jinghong quickly applied the heated sample on the grid. And this is protein on the grid. We think during the freezing process, some proteins got exposed to colder temperature, but it doesn't matter. We can still image them, get 2D projection, and classify them, and separate the different conformations. So this is exactly what we observed from our structure data. You can see the consensus map looks really awful, although the transmembrane domain looks fine. But the intracellular domain in such a poor quality is almost disappeared. But after doing extensive classification, we got two different classes. You can see how different their intracellular domain are. No wonder the intracellular domain is in such a poor quality. So the first conformation, the blue one, is the same as we determined previously at low temperature. And the second one, the yellow one, is a new conformation and has never been determined, observed before. You can see the intracellular domain is tilted in the yellow conformation is tilted towards the membrane compared to the first class. And this results in a shorter and a fatter shape of trypan4. So we name these conformations as cold and warm conformations to this just to reflect the temperature at which they were identified for the first time. It's not necessarily mean that the cold conformation only exists in cold temperature. Actually, in the absence of calcium, the cold conformation also exists in warm temperature, which I will show you later. And very interestingly, we observed a calcium binding site in the intracellular domain of the warm conformation, but not in the cold conformation, because this calcium site this only exists in the warm conformation. We name it calcium warm. And so from this morph, you can clearly see how this calcium warm site is created. In the cold conformation, the blue structure, the coordinating residues are far away from each other and are only brought together to create the calcium binding site in the warm conformation, the yellow one. So you can see temperature is essential for forming this calcium warm site. And to understand the function of this calcium warm site, Songjin mutated a key residue in the binding site E396 to alanine and performed electrophysiological studies. And so in the wild type, you can see the pronounced activation, pronounced potentiation induced by temperature. But in the mutant, the mutant lost the temperature-induced channel potentiation. And consistent with the EFIS data, we determined the structure, and you can see this mutant, the orange structure, no longer bind calcium in the intracellular domain and produced only cold conformation even it's heated to 37 Celsius. And in this structure comparison of a single subunit, you can see the mutant structure is essentially identical to the wild type cold conformation in blue. And with these structure and functional results, we can conclude that the calcium warm site is a key determinant of trypan4 sensitivity, thermal sensitivity. And our data also shows that neither temperature nor calcium alone is sufficient to drive the conformational dynamics of trypan4. It's actually the combination of both factors that synergetically shift the cold to warm transition. And here, let's see the remarkable conformational changes between the cold and the warm conformation. You can see calcium binds to the transmembrane domain site and also the intracellular domain site. You can see the large conformational changes, mainly now focusing on the intracellular domain. This is the bottom view of the protein. Also, we observe this large structural rearrangement of trypan4 at physiological temperature in comparison to lower temperature. Unfortunately, the pore remained closed. It sounds really disappointing, but this also aligns with the EFIS data that the calcium-induced, you can see the calcium-induced trypan4 current is still voltage dependent even at 37 Celsius. And so at this moment, we remembered our old friend DVT. As I mentioned, we previously tried to use DVT to get an open state structure at low temperature. Although we did identify two DVT binding sites, now we call them DVT code 1 and DVT code 2. The pore remained closed. there is a discrepancy between the structure and the functional data. But when we performed the Yifan experiment using the mutant that abolished the DVT binding in those two sites, it becomes clear because these two code sites are functionally irrelevant because you can see the mutants here at this site, they didn't affect the DVT's modulation effect. They look exactly the same as the wild type. That means the DVT just bind to those positively charged cavities and didn't play a functional role. So motivated by the newly identified worm conformation and also the calcium worm site, so Jinhong decided to revisit DVT and determine the trypan4 structure with calcium and DVT at 37 Celsius. And again, he found both cold and warm conformations. The cold conformation, the blue one, still has two DVT cold binding sites, but you don't see them in the warm conformation. Instead, in the warm conformation, it has a robust DVT density in a different cavity. It's located in the interface between the transmembrane domain and the intracellular domain. Because it only exists in the warm conformation, we name it a DVT worm. So to assess the functional relevance of this DVT worm site, Songjin neutralized the charge on each of the 7 positively charged residues in the binding site and performed Yifan recording. And he found all the mutants became insensitive to DVT. Here, I only show you the result for one of the mutants. Therefore, our data supports that the DVT worm site identified at a physiological temperature is a truly functionally relevant site for voltage modulation of trypan4. And even more excitingly, the pore finally opened. The pore is widely open in the presence of calcium, DVT, and physiological temperature. You can see on the left, without DVT, the pore is closed. And on the right, the 4 DVT molecules pulls the S4, S5 linker and the pore line S6 helix through charged interactions, therefore resulting in channel opening. And here is a morph showing the transition from warm closed conformation to warm open conformation induced by DVT at a physiological temperature. You can see the large conformational change throughout the protein. And this is a bottom view of the protein. And this is a zoom-in view of the transmembrane domain. You can see how the DVT molecules pull the pore line S6 helix to open the channel. Still have time? Okay. Next, we extended our studies to a third type of ligand, ATP, which is an endogenous trypan4 inhibitor. So at the room temperature, as I mentioned, ATP effectively inhibited the calcium-induced trypan4 current with an IC50 in the micromolar range. This is actually very puzzling because on the cellular conditions, the concentration of cytosolic free ATP is a lot higher than IC50. It means that ATP, the trypan4 would be inhibited all the time in the cellular environment, which is kind of strange. However, when we did the same experiments at 37 Celsius, we observed a substantial reduction in ATP inhibition, as you can see here, with less than 50% of the current inhibited even at millimolar concentrations. And this result indicates that the ATP is actually not a potent inhibitor for trypan4 under physiological conditions. So Jinhong further determined the structure of trypan4 in the presence of calcium and ATP at 37 Celsius to explore the possibilities that ATP may also have a temperature-dependent binding and inhibition. So again, he got two confirmations, cold and warm. And more importantly, we observed that the ATP indeed occupied different sites in these two conformations. They are nearly 20 angstrom apart. So I will skip a couple slides and go to the last one. So the key takeaway message is temperature matters. Proteins like trypan4 may adopt different conformations at different temperatures, affecting how they interact with ligands. With this, I hope that I can convince you studying proteins at right temperature whenever possible could be very important. And after we published our results, people have been asking, what does that mean? Does it mean we need to determine all the structures that were determined at low temperature, or should we redo all the EFIS that was carried out at room temperature? We said, no, this is not what we mean. We mean if you see any discrepancy between the structure and the functional data as we observed for trypan4, temperature could be one of the very important factors to be considered. And so in the end, I want to thank all the lab members many of them who moved with us to Northwestern and rebuilt the lab. And also want to highlight the important contribution of Jinhong and Xiongjing for their amazing contribution for the trypan4 temperature study. And also want to thank all the private foundations and NIH for supporting our research. Hopefully, NIH can continue supporting our research. Thank you very much for your attention, and I'm happy to take any questions.
Thanks, Jen. This is wonderful. We have a couple of questions, but some of the questions are addressed to both you and Sasha, and I will leave those questions to the end of after three speakers. I do have actually one question that you mentioned that in the when you warm up the temperature, you have both conformation, and then in your talk you kind of implied that the cold conformation was kind of being trapped during the plunge freezing. But I wonder whether this is also related to basically this conformation has kind of ensemble biased by the temperature rather than being cold trapped during the plunge freezing.
It's possible. It's really hard to tell when is those cold conformation coming from. It's possible to it's just dynamic between the cold and warm, even at 37 Celsius, or some of the protein get exposed to the cold temperature during plunge freezing. We cannot tell.
Yeah, and one of the questions from audience is that what is the hypothesized function of ATP binding?
Oh, the inhibitory function of ATP?
Yeah,
inhibitory mechanism?
Yeah.
So because of the matter of time, I didn't clearly explain that. We think the ATP binds to trypan4 to stabilize trypan4 in a warm closed conformation or warm cold conform the warm closed conformation or cold closed conformation. It cannot move to the next step to the warm open conformation.
I see. Cool. Let's say pause it here and then pause into the our final speaker today, David Julius. It was addressed to Sasha and Juan, but it's probably also related to all this trip channel. So the question is that these channels are not just floating around freely. They're often anchored on the cytoskeleton by the C terminals. And then the question is that the connection is absent. Does this one influence the certain temperature sort of sensitivity of the channels? And I actually don't know, but certainly they interact with many others soluble components. But I actually don't know how does it interaction with the cytoskeleton.
Yeah, I think well, I'll chime in quickly, and then I'll let Sasha and Juan say something. I think that's a question I think that can be, we're looking at channels in membrane, in cellular membranes can probably be more instructive than nanodiscs or detergents, where you've first had to excise the protein from the membrane with detergents. So that's one promise there. I'd say this brings up a more general question that sort of vexed the TRP channel world, which is that we know that there's a large part of these channels in terms of mass that protrudes into the cytoplasm, and many of them have some very interesting folded regions, like in TRPM8, some of the TRPM channels use different domains, MHR domains, etc. We still know relatively little about what those do. They're highly conserved. In some cases, well, Juan's talked about some interactions with ATP and things like that, but in many cases, we don't know what these protein folds do. We don't know what they interact with. TRPA1 is another example where there's this huge part of the protein that consists of these multi-repeat anchor and repeat domains that hang into the cytoplasm like a huge skirt. We've always presumed they must interact with the cytoskeleton or other proteins. We still really don't understand what they do. On the other hand, I can say that you can see temperature sensitivity for many of these channels in excised preparations, as we did years ago for TRPV1 with function, where you can reconstitute them into, well, you can just look at them in detergents or reconstitute the membranes where you can see temperature sensitivity. So baseline temperature sensitivity may not require those interactions, but it could be very well modulated by things that we don't yet understand.
Yeah, I can add only kind of to one way of addressing it would be actually going into cryotomography, right? As David mentioned, right? More natural preparations, or like taking natural environment and trying to preserve it in our imaging techniques. So, and of course, we are not, I guess, yet there. The resolution of cryotomography for such relatively small particles is not that high, but eventually, hopefully, we'll be able to visualize these systems as a part of the environment and see these mentioned anchor proteins connected and membrane with other components, modulating and affecting temperature sensitivity and other functions of these channels.
Ryan, you have anything to add?
No, I don't have anything to add.
Cool. And I think this also really related, like Sasha said, I agree that technically, either by nanodisk purified protein reconstitution or by vesicle approach, they all have to really remove a lot of like a particular interaction with cytoskeleton. And then those really only able to be visualized when you look at an all entire cell, intact cell, by probably tomography and other things. And then certainly at that level, that the limited resolution probably only allow one to, in combination with a high resolution structure, and then to try to understand that. That's the next, really the challenging, and then the technically a next step for starting all the channels. Yeah, and I don't also, the other thing is that, do you guys have questions about each other? So I think it seems like we answered all the questions listed there, and that's amazing within the time frame. And but unless you guys have a question about each other, that's also your.
Yeah, so I have a question for David. Beautiful talk. So I'm just wondering, you saw the different temperature sensitivity of TRPM8 in different species. Do you think this is related to their pain sensation among different animals?
Yeah, well, that's there probably have not been a lot of behavioral studies on penguins and other things like that, so it's kind of hard to address. Actually, there hasn't been that much in the old days, people did do a lot of recordings for different types of stimuli from chick sensory neurons. I should go back and look at that, but I don't think there was a lot of analysis of temperature sensitivity. It was mostly for chemical sensitivity. So, we don't really know. If you think about birds, they're often, at least some birds that live in the cold, like penguins and stuff, they do have the, very well insulated feet or talons. and they do have, they are protected against, a lot of these environmental stresses. But I don't really know the answer to that. I should also say that if you look at fish, like if you look at zebrafish, they don't have a TRPM8 ortholog. They have a couple of different TRPA1 orthologs. So there is a big change in some of the distributions, and certainly in the threshold of activation of these channels phylogenetically. And we could only presume that that relates to behavior, but I think the behavioral analysis there is woefully inadequate to really make conclusions about what the relationship is to, say, thermosensate, core body temperature regulation versus nociception.
Cool. Any other?
I have one.
Yeah, please.
So I like thinking about amide exchange, and I noticed that you'd mentioned about the distributed nature of these changes. And I'm just wondering, you have a beautiful mechanism, a beautiful site that's really critical in controlling transitions between different states. But I'm just wondering, do you have any clues from anything else about the distributed nature of the interactions that are involved in these switches or these changes in states?
I guess the question can be for all of you, right?
It is.
Yeah. So.
Well, I think they're distributive. I think it is clearly distributive in that multiple things are moving. I think, what Xiaoxuan's analysis, HDX analysis shows is that there are regions that are more energetically temperature labile than others that account for temperature sensitivity. And in the case of the mammalian TRPM8, that's, likely and actually consistent with mutagenesis studies from our lab, from your Grandel's lab, from Artem's lab, that, have identified, that pore helix region in the outer pore loops as an area that's where you do get, you get mutations in various parts of the protein that shift thermal threshold, but that seems to be one of the hot spots. And so I think in the case of at least, to some degree, TRPV1 and TRPM8, that's a region of the channel in mammals. I think that's a region of the channel that's moving a lot. And one thing I didn't mention is that, one of the things that's been challenging for TRPM8 is that many of the early structures that we in the Lee lab got, you have trouble seeing high resolution structures in the transmembrane regions, which we always presumed is because, especially around cold temperatures where you're working, that area is very malleable and moving. And I think that in fact turns out to be the case. And we think that's the case because if you look at the bird channel, when it's locked, you can see better resolution there. The mammalian channels have been particularly difficult to work with, although now in these different preparations, you can see it better. And with the bird channel, you know what you're looking for, so you can be on the lookout for those things in these structures that previously, as Sasha said, we would sort of put aside because they weren't high resolution. So I think that region is energetically and conformationally quite dynamic, and that is a key area for driving thermosensitivity. So it is distributive, but there are regions that I think are hotspots energetically.
I can only add a little bit to this. What I was talking about, the conformational wave, right, which covers big chunk of the protein. So we've been just comparing different states, right, because the conformational changes are so distributed. We've been trying to see if we can pinpoint specific interactions which would characterize transitions between these states. And actually, that was quite challenging because of so extensive region of movement. We have actually many bonds are getting broken or reformed, right? And so it's very hard to make a sense of like what are more important, what are less important. Of course, many of them van der Waals, but many of them are hydrogen bonds and ionic interactions as well, which are like contribute more energy. And even those, like many of them, and we've been trying to use some prediction tools like calculate the overall number of bonds, and that's enormous. So it's hard to then choose through like, understand like the complexity, and then just by eye, just, selecting with, your hands and looking at specific interactions also I found myself kind of lost in the complexity. So it's many molecular interactions are changing during these transitions, and that's because they're quite distributed.
I should say that there's a nice paper on a preprint from Feng Chin's lab, where he's recently looked at structures of TRP1, where he suggests that there are these local protein unfolding events in 3 or 4 regions that he calls, I think he calls them latches, I can't remember, around, at key areas like around the TRP domain and others that he thinks undergo sort of a, ordered kind of local unfolding. Whether that unfolding is reversible or not, I don't know if he knows, but his hypothesis is that you get that protein unfolding is what drives this process more so as opposed to, say, changes in heat capacity, which our results would, I think, hone more to the heat capacity model. But in any case, he does map has some nice data to map these local changes around areas that are sort of, transition points around the pore. And so, he may have zeroed in on some nice areas for TRPV1 that are kind of hotspots for temperature sensitivity.
Yeah, for us, I fully agree with what David and Sasha said. The temperature induced dynamics is likely distributed throughout the protein. But I also want to emphasize that we have observed quite strong dynamics in the intracellular domain. In addition to the transmembrane domain, we have seen these dynamics in the TRPM4, TRPM3, and TRPM2. And TRPM5. And so we don't know if these are the real sensor, but we think they should be involved in this kind of temperature dependent conformational dynamics.
Thanks. And I see that also what Jen mentioned, that also another implication is that probably for not only for membrane protein, a channel, many other protein, even intracellular protein, their function is could be really influenced by the temperature. And this is probably another next frontier or another area that for structural biologists to think about it and really take into the temperature into the consideration when they study structures or interpret the data. And we think we're end of the perfectly 2-hour time limit. And I want to thank everybody, all the speakers, wonderful session, and also thank all the attendees and then to listen to this wonderful talks. Thank you all. I think we end this webinar now.
Okay.
Thank you.
Thank
Thanks
you, everyone.
everybody.
Thank
Great.
you, everyone.
Everyone.
Thank you.
Thank you.